A simple, versatile, and low-cost in vitro hydroponic system was successfully optimized, enabling large-scale experiments under sterile conditions. This system facilitates application of chemicals in solution and their efficient absorption by roots for molecular, biochemical, and physiological studies. We demonstrated that this system was highly efficient in obtaining homogeneous seedlings along initial development, as well as separating roots and shoots using Arabidopsis thaliana and Setaria viridis. The following items are necessary to build up the hydroponic system and cultivate plants under sterile, controlled conditions. 7% ethanol, 10% bleach solution, polysorbate 20, MS medium including vitamins, MES monohydrate, agar, potassium hydroxide. Laminar flow hood, hot plate, growth chamber. Disposable plastic boxes, sterile glass Petri dishes, adhesive tape, sterile 200 and 300 microliters pipette tips, scissors, 300 microliters multi-channel pipette, 200 microliters pipette, scalpel blade, tweezers, and 200 microliters pipette tip racks. It's crucial that the pipette tip flat has an area to be filled with the solid medium. Sterilize the pipette tip racks without covers that will be used as mini tanks in the autoclave at 121 degrees for 20 minutes. In order to avoid contamination, all the previously mentioned materials were sterilized in the autoclave.